Good evening, everyone. Welcome back to 461 review weeks, and today we're going to be going over essay question 1 for Unit 2 for talking about muscles and all of that great jazz. So we have a lot of material to get through tonight and not a lot of time. So I pre-drew some drawings that we're just going to go through together and talk about how they work. So what we want to start off is kind of the basis and the makeup of muscle and how that kind of works all together. And it's super interesting and super cool because at the base of our muscle fibers, and specifically in our skeletal muscle, we have our base unit that's going to be called our sarcomere. Sarco means muscle, and when we're really, if you zoom in super closely on a muscle, this is what you're going to find. This is what's going to contract and this is what's going to move, allowing your muscle to contract. So there's some different components to this. We have, as you can see, I drew up a little part already, but we're gonna need to add some other components to this. There's three main parts to this. Three main moving parts, I guess I could say. Right here, this is gonna be called the sarcomere. It's a little like this black line is called a Z-disc. And Z-disc to Z-disc, so just like this little area right here, is going to be called that sarcomere. There's going to be thousands and thousands of sarcomeres between each muscle, in each muscle fiber, and so that's what is allowing your muscle to contract every time that you lift something and move, and all those great things. But right here, in the makeup, kind of like the scaffolding of our sarcomere, pardon me, is we've got these black lines right here. This is going to be called our nebulin. And then up here, these little pink guys that I drew, these are going to be called our titin. Titin's going to be more of like a springy structure that's going to help us move some of our moving components that we'll talk about in just a second. So to add on to this, we need to add things that can grip and move and pull and push and cause the kind of that contraction to occur. So let's talk about that. We've got one structure that we're going to call actin, specifically G-actin. These are going to be little ball-like structures that have little ATP binding sites on them, all of those things. And when we line it up in a form, we're going to see just lots of little G-actin lined up, actually specifically on the nebulin, chilling all together, looking fabulous. And all of their all of their ATP binding sites are going to be pointed out. All their ATP. Well, places where ATP is stored. Because that's also a binding site for another thing that I'll talk about in just a second. Not to be confused. Right there. That's kind of what we're looking at. So if I was to draw it over here, it would be like boop, boop, boop. But I'm going to simplify and do a little like this. This is what I'm drawing. Like so. Absolutely so anatomically correct. Then we're going to add a little new character called tropomyosin. Tropomyosin is going to be kind of like a little rope-like structure. And tropomyosin, I think it's P-O, trope. Tropomyosin actually is going to cover some of these binding sites right here to prevent contraction from occurring all the time, which is really important because you don't want to be constantly in contraction. Because what binds to those sites is actually going to be our myosin. So these little areas that store ATP right here, are the spot for ATP. We don't want those to be bound to myosin all the time, and so we cover them with that little rope so they're not bound to ATP all the time, which is really important. Now, we can't just have that rope covering it and then expect it to be moved without us moving it, right? So we have another structure, very important structure. Actually, also very important to note is that when all of the G actins are roped together, this is going to be called F actin. Filament actin, which is gorgeous. Now, and then this little thing is the myosin binding site. Anyway, beautiful. Moving forward, last part of this is that we have a gorgeous structure called tropomyosin. Tropomyosin is going to be a 3-part structure, little Mickey Mouse, if you will. And this is going to be our troponin. Lots of T's here, but troponin, tropomyosin, very different. This is going to have 3 components. We're going to say troponin C, troponin T, and troponin I. Now, for all of these, we're going to go with T, I, and C to describe them all. Each of them are going to have different functions and properties that are going to help us do different things within the sarcomere, right? So, well, specifically right here. Troponin T is going to move tropomyosin. Troponin I is going to bind to our G-actin. And then troponin C is going to bind to calcium. It has 4 binding sites for calcium. And what's cool is that since there's 4, 2 of them have high affinity for calcium and 2 of them have low affinity. And affinity just means that it has a strong desire for it. And so it means that 2 of them will always be taken up. Well, two of them are not always, but when you have more calcium in there, they will be taken up, which is very important. That's why having more calcium will cause more contractions. Beautiful. So there's a little tropomyosin, and that's just gonna be on here, right? And that's what's going to then move that. Troponin's gonna be on here, and that's what's gonna move that rope of tropomyosin off of our actin filament for it to be exposed. So if I was to draw it over here in our sarcomere, we would add in our tropomyosin right in here, and then we would just add in our little Mickey Mouse troponin structure, and it would be on our bound to our G-actin proteins, which is fabulous. Now, what are they trying to move the tropomyosin away for? It is going to be, they're trying to move it for that myosin binding site, so you'd assume it would be for something called myosin, right? And myosin is super important and super cool, and it actually has a cool structure. It's going to be more of a heavier chain, so it's going to, if we look at a actual histological slide of it, it's going to look more dark and heavy. And so we actually call it the dark band versus the actin. We look at the light band, which is cool. And what's cool is when we see it, they kind of just they look like little cocked heads like this, where they have little spots to come up and bind to their little sites right here, which is super fabulous. And just looks like a bunch of little centipede legs, but it's kind of cool how they can do this. Now, properties of myosin here are that they have some heavy chains together, and then they have heads, like so. And they have, so this would be their tail right here, and then they have this hinge area, and then they have heads right there that would separate out into different areas of the sarcomere, different actin sites, which is super fabulous. Now, we have different positions for the myosin, and we have either a cocked position where it's ready to go, ready to be, ready to bind to its myosin sites, or we have an uncocked position where it's already been bound and already moved the filaments along. So that's a little bit about our sarcomere. It really gives us the ability to move, and then, as you can imagine, when a myosin would bind to its myosin binding site, it would cause a contraction to occur because the muscle would then shorten and come together, and that would cause contraction. And that is because of calcium entering the cell. Now let's talk about how that happens. How does calcium enter the cell to allow this whole contraction to occur? Well, it happens because of something we call the neuromuscular junction. We need neurons over here to bring that action potential forward so that so your muscle knows that you want to move. Because this is a voluntary movement, right? But you have to know that you want to move. But pretty fascinating. So here we start over in our neuron, and we're going to end at like the terminal end of the axial end of the neuron. And we have a few channels down here. And here we've got our trusted bestie, our voltage-gated sodium channel, as well as a couple other channels like our voltage-gated potassium channel, and, of course, our voltage-gated calcium channel. Because calcium is super important for contraction, we need that. But calcium is also super important for something else. Calcium is super important for exocytosis. Calcium drives exocytosis. And what we need to exocytose here is a neurotransmitter called acetylcholine. Here in that axonal terminal of this neuron, we have little vesicles full of acetylcholine. And acetylcholine is going to drive this action potential right here. So we've got vesicles full of acetylcholine, and on each vesicle we've got snare proteins. And snare proteins are cool because on each vesicle the snare proteins are there, and then on the membrane we have snare proteins. And with the help of calcium entering when we have that action potential sweep down the neuron, it would open up these voltage-gated channels, and when it gets to the calcium one, calcium would flood in, and calcium would bind to both of these snare proteins, and it would bring them together, causing a conformational change, and they kind of twist, and then that allows this vesicle to merge with the surface of the membrane, causing exocytosis, which is something we've already talked about in previous units. But then, as you can see here, we would have exocytosis of this neurotransmitter, and in this case we're going to use acetylcholine here. And acetylcholine is going to then act as a ligand or a key to open up some channels here on the surface of the membrane of this muscle cell, the sarcomere here. Sarcolemma, if you will. That's going to be the surface of the membrane of the muscle cell, so the sarcolemma. And right here, we're going to see it come down and act as a ligand for our nicotinic type 1 channels. Beautiful. That's going to allow positive ions, specifically sodium in this case. Sodium is going to flood into the cell. And we're going to have a little bit of potassium leave, but the driving force there is going to cause a little bit of an action potential. It's going to cause an action potential to occur. And this is just another nicotinic type 1 channel, but we're going to see that boing happen along the surface of the membrane. And before we move on, it's important to note that the acetylcholine needs to be used up or eaten up. And so we're going to have this little enzyme that is going to kind of break down the rest of that acetylcholine. And they've named it really beautifully. It's just going to be called acetylcholine esterase. I'm going to abbreviate, which literally just means it breaks down the ester bond in acetylcholine, which is beautiful. I drew it as Pac-Man. And that's beautiful so that we don't have leftovers of acetylcholine to continue that stimulation. So once that signal has been sent, we want that signal just to be very brief. And so once that signal has been sent and received, we use up the rest of that neurotransmitter, which is why you don't continue to contract after that signal has been sent. So that action potential will be sent down the sarcolemma. And what's great is that just, your muscles are very complex and very thick, but just like cooking a potato in the microwave, sometimes you just need to stab holes in it to get that heat down in there, and sometimes we just have to stab holes in muscles to be able to get the action potentials in there. So we have some different holes in our muscles called T-tubules. Which just stands for transverse tubules. They just, run across transversely. And this allows our action potentials to actually continue to sweep down the membrane here. And we continue to see different voltage-gated channels open and be stimulated through this. So I'm going to zoom in a little bit on this. But here, this might actually just get a little bit small. But here we can see that there's still, I just drew them according to the same colors. But still we're going to have some voltage-gated sodium channels, voltage-gated potassium channels, and we're going to have a voltage-gated calcium channel, but we're going to call it a different name. It's going to act differently than just a voltage-gated calcium channel. It's not going to act as a voltage-gated calcium channel. We're going to call this dihydropyridine, and we're going to just abbreviate and call this DHP right here. DHP is going to be really cool because it's going to act with its friend right here. We're going to call that RIR, or ryanodine. And ryanodine is also going to connect, sorry, this wasn't the best drawing, but it's going to connect right here with our sarcoplasmic reticulum, or our smooth endoplasmic reticulum, but since, again, we're in the muscle, we're going to call this sarco for muscle, our sarcoplasmic reticulum. And this is incredible because this is where our calcium storage is found. So calcium is just found in here in a plethora amount. And remember, calcium is what's going to drive our contractions of muscle. So let me make this a little bit more correct. Ryanodine's connected to this. So once our action potential, or BWOG, sweeps down our membrane, it's going to stimulate DHP and ryanodine. And what's beautiful is that we have this whole instance where it's kind of sodium-induced this reaction right here, but we're going to have that calcium release for this. And so calcium is going to be released through ryanodine, and it's going to come out into this sarcomere right here, which would be out here, and it's going to be out in here in the sarcomere. And that's how it gets out there. Now we're out here in the sarcomere, and now we need to talk about how that contraction actually occurs. So if we zoom in here on this little weird circle that I drew over here, we're going to talk about how that contraction actually occurs. We're going to call this the cross-bridge cycle. We've got 4 steps that we're going to specifically talk about. And it's super awesome because we just need that calcium to come in for this to actually work, which is beautiful. So step 1, we've got our myosin head with its ADP binding with its ADP on there, and it has a little phosphate group on there. And what's beautiful, or wonderful, amazing, is that we have some calcium that comes in. Didn't mean to do that in red. We have calcium that comes in, from this whole neuromuscular junction right here. It's going to come in, and when that happens, we're going to see a little change take place. We're going to see our whole thing is going to come up and bind to our actin, and specifically on that myosin site. And when this happens, we're going to just release some heat right here. It's going to release some heat, which is so super fabulous. A little reaction right there. And then we get to have the power stroke happen. And the power stroke is when we get to see a change occur where it's actually moving and shortening the muscle. So this is going to be the power stroke. What's going to happen here is that we're going to release this phosphate group, and we're going to release this ADP. So what happened here is whole thing right here. Let me make this super correct. We then, you can imagine, pulled that muscle forward, and that's going to be our power stroke. And then we also are going to release some energy there, right? That's what's going to cause our contraction right there. That's what's going to happen. And next we're going to then enter in some ATP, and this is where we're now going to continue to be in that cocked position because we just moved, and we'll again release some heat. They just are always releasing heat. That's why you get hot when you exercise. But this is what's going to, oh, sorry, I forgot. When we're still in cocked position, but we're releasing from that myosin binding site because of ATP. ATP has to come in for us to be able to release. When we don't have ATP, then we can't release from the actin. Then we're going to go through some hydrolysis to be able to get back to our original, because we still have the 3 phosphates, but they're just separated now. So we have to get back to our original form. And that is what happens in skeletal muscle. So we have some different types of muscle as well. And it's important to know how they are different. So we talk about 3 types. We have type 1, type 2A, and then type 2B or type 2X. I don't know why there it says 1, but 2. Now, let's talk about all the differences here. First, if we talk about the difference in our circuit pumps, type 1 is going to have very slow circuit pumps. It's going to be very different from the rest because our type 2 is going to have like kind of medium circuit pumps, and type 2B is going to have very fast. We'll see how they use energy is that type 1 uses aerobic, type 2A uses both, and type 2B uses anaerobic. So type 2B, if we're just using those ones, it's hard to go a long time without oxygen. Now, for how fast they fatigue, type 1 doesn't fatigue. It does not. I'm going to put nada. Type 2 is very resistant, but it still can fatigue. And then this one is very fast to fatigue. So, if we're keeping track, type 2B, you need lots of oxygen and it fatigues very fast. Now, for our blood supply, type 1 has lots of blood supply. This one has a medium. And this one has little. So it's even harder to get oxygen there. For myoglobin, these 2 both have it, and this one doesn't. And then the color, it goes with that, but these ones would both be red, and this one's white. So those are some differences between those. Type 2B is mainly found in cheetahs. That's why they can run so fast for short periods of time, but then they have to rest because they can't keep up. And then type 2 is going to be found in like more elite athletes. They still have type 1, but like humans mainly have type 1, but type 2 with like athletes, like long distance runners and stuff like that, because they can continue to run and not be fatigued. And then type 1, all humans just have mainly type 1, but we actually have a mix of all 3, which is very cool. And then if we go to our energy sources for how we are getting this, how we are getting either anaerobic or aerobic. For anaerobic, we have a few different ways. We have cytostolic, I forget how to say that word, but let's write it out. Cytostolic. ATP. I don't know why that was such a struggle, but that's okay. And this takes about 5 seconds for us to create. Then we have creatinine phosphate, creatine. And that takes about 10 seconds. Then we have glycolysis. Which takes around 1 to 2 minutes. Then for our aerobic oxidation, this is going to be our electron transport chain. And this can last for like hours, which is great. Now, we're going to go back and kind of compare our skeletal muscle cross bridge formation to smooth muscle. And then we'll move into our reflexes, and that should end our night. Now, okay, so very different because in our smooth muscle, it's not as organized as skeletal muscle. It's and we don't have those sarcomeres, and so we need to have something else to be able to help us keep track. Because specifically, actually, our main difference there is that we don't have troponin in smooth muscle, and so we need to add another phosphate to be able to make those contractions actually happen, to be able to actually move tropomyosin away, because we don't have that troponin, if that makes sense. But what's great is that smooth muscle is mainly found in our intestines and stuff like that, and so it's going to perform things like peristalsis, or like that smooth muscle contraction that continues to just flow, right? And so we're going to have those muscles that are going to continue to contract, and also in like sphincters and stuff like that, in your muscle or in your like stomach and stuff like that, that will continue to stay contracted so that you don't have to think about that. And we need a way for that to happen, if that makes sense. So let's talk about some key differences here. We have 2 different types of contraction in smooth muscle. We have a phasic and then a tonic. And there's a key difference here for this. But first, for smooth muscle, let's talk about phasic. Phasic, however you say that. But same thing, the thing that still drives it is still going to be calcium. So we need calcium to come in for this. But we're going to have a new thing come in. This is going to be called MLCK, along with ATP. Give us some energy. Now, when we do this, we're adding in another phosphate group, which is great, because this is what's going to allow us to move that tropomyosin out of the way. Now, once we kind of reach this phase, we do still need to remove a phosphate so we can bind. So we remove a phosphate. Kind of seems counterintuitive, but I promise it works. And then we're also going to remove our ADP. So here we go, we remove a phosphate, and we also remove our ADP. So we're just left with our myosin head, and we go up, make that power stroke and cross bridge formation, and then we're going to add in some more ATP again, and this is what's going to give us the power to uncock that, or not uncock, but like uncross-link that myosin head. But we still have that phosphate attached. Now we have a new protein that we're going to add. And this is going to be called MLCP. Different than MLCK, because MLCP is going to allow us to remove another phosphate. However, MLCP breaks the cycle. It doesn't always come in. This is what's going to get us back to here. If MLCP doesn't come in, then we can just stay and continue to make these contractions, and this is why we could continue to have that closing of the sphincters and stuff like that. Now, in our tonic contractions, this is going to be a little bit different. We're going to actually put in that protein a little bit sooner to change the state of this contraction. So we're still going to have our calcium come in. Same thing happens, MLCK. ATP. Same things. This should all be very familiar. Phosphate comes in, or sorry, our phosphate comes out, and then ADP comes out. However, then you can see that our phosphate comes off, and because our MLCP came in earlier right after we did our power stroke or our cross bridge formation. And this changes because this causes us to stay in a latch state for much longer. So then we get our ATP. There's nothing keeping us, and then we can continue to just have like a change of ATP in there, and then we just stay in this latch state forever, which is great. And that's our smooth muscle differences there between skeletal. So very different. Having some new things in there that allow us to stay contracted and aren't as busy. Now for our reflexes, we just have a few things with this one. And it just has to do with our receptors and what we're looking at. Now in our muscle fiber, or in our muscle, we have a few different types of neurons and different pathways, but we are just going to specifically look at the different parts of the reflexes, but we do have a muscle spindle within our muscles that contain a bag fiber and a chain fiber that allow us to sense when we're stretched. And so that's going to have to do with our stretch reflex down here at the bottom. I drew out this diagram already. And as you can see, our stretch reflex is basically just allowing us to prevent overstretching of our muscles and our tendons. And so it's just to prevent overstretch. Which is great. And we're going to use our sensory neurons through our tendons right here. So this is just like our patellar tendon. And this is classic, like you just get your knee hit at the doctor's office. It's going to stimulate contraction of your quads, right? And so that's because if we stretch this tendon, like you got it hit with a little hammer. Here's my little hammer. Boop. Then we're going to be sending a signal through that sensory neuron, a little EPSP, to our spinal cord. Now, this is where it's super cool because we split in our spinal cord to send signals back out. Since we are sending reflexes, we don't go all the way back up to their brain, and we're just going to we're going to use the spinal cord as our control center. And here we want to activate one muscle and like stimulate another muscle to not contract, right? And so we're going to have a little interneuron here that's going to send an IPSP. And that's going to allow our hamstrings to relax, and then our quads to contract so that we can respond by contracting our quads to not stretch that tendon, which is great. Another cool reflex that we have is the Golgi tendon reflex. And as you can see, this man, definitely Brother Hunt, is lifting 100 pounds, and he can really feel it on his tendon. It's stretching his tendon. And so his tendon is responding, and the goal of the Golgi tendon reflex is to prevent overtension of muscles and tendons. And so your tendon, the Golgi tendon organ inside of the tendon, is going to send a action potential, specifically an EPSP again. And again, we're going to go to the spinal cord. We're going to have interneurons, and your interneuron is again going to send an IPSP. And now we want to make sure that we relax our so for example of our bicep, if we were contracting that and we were causing too much tension on that tendon, we want to just drop that weight and relax our muscle, and then contract the opposite or antagonist muscle that we're going against, which in this case would be the triceps brachii. So that's what would happen there. The last one is going to be the withdrawal reflex, and this is just to prevent further tissue damage. And this is like a little kid touching a flame, and you move your hand away because you don't want to get further tissue damage, which makes sense. So the receptor here is actually just going to be nociceptor sending pain signals. So it's not specifically any crazy signal, but it is an APSP. You're sending a signal. And then here in the spinal cord, again, we'll be sending an IPSP to the opposite muscle, the antagonist, and then you'll send that EPSP to that agonist muscle that you want to contract to take your arm away, and that you can scream at the same time. And those are all your reflexes that we'll go over today. Now, brief overview of everything we went over. We talked about the sarcomere and what makes up the sarcomere with its different components like G-actin, tropomyosin, troponin, and how all of those work together to be the kind of scaffolding with nebulin and titin for the sarcomere. And then we talked about how the sarcomere fits into the neuromuscular junction and how we can get calcium into the sarcomere to cause a contraction. Then we talked about how a contraction actually occurs in skeletal muscle, calling it the cross-bridge formation. We compared that to smooth muscle with different types of smooth muscle contraction like phasic and tonic. We talked about the different muscle types throughout our body, like type 1, 2A, and 2B. Talked about where we get different energy sources, and then we talked about all 3 different types of reflexes and how our muscles respond and react through our nerve stimulation. And that was it. So that's going to be our essay question 1 for Unit 2, and hope you enjoyed listening. Thanks so much. Have a great night. Bye.